Welcome everybody to the lightning talk session of this third MGI Symposium. Delighted to be here. I'm Mike Benke from the Department of Biostatistics. Since it's lightning talks, I should make the introduction short to let people have as much time as they can possibly have. Our first speaker is Aleda Leis, research investigator in epidemiology, who will be speaking on polygenic risk scores for cardiometabolic disorders in association with postoperative acute kidney injury.
Great, thank you. So briefly to start, postoperative acute kidney injury is a significant postoperative complication. It's characterized by a reduction in kidney functioning within 7 days post-op. There are many risk factors for AKI, including cardiometabolic disease and genetic factors. There have been a number of studies examining candidate genes for increased risk of AKI, many of which have variants that are related to the development of cardiometabolic disease and inflammatory processes. So the objective of this study was to examine the association between polygenic risk scores for type 2 diabetes and postoperative acute kidney injury. There's the potential and the thought that high polygenic risk scores could be used as a proxy for subclinical or undiagnosed conditions, in addition to potentially having the phenotype. And additionally, ICD codes alone may not fully capture complex conditions like type 2 diabetes and often underestimate the prevalence. So there's the potential utility for polygenic risk scores to be used where these conditions might be covariates of interest, but otherwise might not be well captured. So briefly, our study inclusion and exclusion criteria are shown here. To avoid upweighting the polygenic risk scores of individuals with multiple surgical cases, only the index case meeting all inclusion criteria was included in the analysis. We also excluded those individuals presenting for cardiac, renal, urologic, and liver transplant surgeries, as the mechanisms for AKI differ within these populations. This leaves a final sample size of 14,824 patients. The polygenic risk scores were taken from studies already published in the literature, and the criteria for potential polygenic weights were shown here. Generally, we looked at studies that had a large external GWAS that accounted for linkage disequilibrium and computation of the weights. Those were derived in European-only cohort, available for Bill 37 of Human Genome, and published in a peer-reviewed journal. We constructed two scores that were published in the Mars 2020 study and the Mahajan 2018 study. And both scores showed about a 1.9 increased odds of type 2 diabetes for one standard deviation increase in score, so they had pretty good determination of the phenotype within our cohort. Statistical methods, very, very simple. We just did logistic regression modeling, looking at the highest decile polygenic risk score against the lowest decile, adjusting for preoperative and intraoperative factors against the outcome of acute kidney injury. The forest plot here shows the model results comparing the highest decile of the polygenic risk score for type 2 diabetes to the lowest decile. Each row represents a different model. Statistically significant effects are shown in red. And as you can see, both the phenotype itself and the Mars polygenic risk score showed statistically significant increased risk of AKI. While the Mahajan score did not show that statistical significance, there was a little bitty trend in that direction as well. Significantly, though, for the phenotype in the Mars cohort, there was only a 5% attenuation from that of the diabetes phenotype itself, and the C statistic was pretty good for all models. So in conclusion, we found that the highest decile of the polygenic risk score was statistically associated with an increased risk of AKI similar to the phenotype. Additionally, we found that there was minimal changes in population level model area under the curve between the phenotype models and those including polygenic risk scores. And additionally, other model covariates, which were not shown, did remain fairly consistent regardless of whether the phenotype or the polygenic risk score was presented. So this indicates the potential utility of using these polygenic risk scores in models where the condition of interest might be not as available as genetic data to create polygenic risk scores for these conditions. Thank you.
Thanks very much. Questions? So that was really good. So you went through it quick, but so how much did vascular injury or hypertension contribute to it? If your multiple linear regression that you put up there, were other things really more related to it or less related to the acute kidney injury, in addition to the polygenic risk?
Yeah, so in these models, both hypertension and vascular injury, we kind of only really had coronary artery disease as a comorbidity. Both of those had fairly large effect sizes. I don't remember what they were off the top of my head, but they also were, as you would expect, statistically significant in these models.
So I was actually thinking about the comparison of the two PRSs a little bit differently. Could you sort of speculate like how those two PRSs are different, and maybe the differences might tell you something about the sort of pathophysiology they show?
Yeah, so I think the biggest difference from when we ran this data was the one did have a slightly higher, let's see, did I have that? Yeah, so the C statistic, you know, and the odds ratio for diabetes were pretty similar between the two. However, especially in these higher deciles, you can see the blue line here for the Mahajong score wasn't quite as high for the percentage of those with the actual phenotype. So I think there's potentially some confounding there where the score might not have been predicting as well for those individuals within our cohort compared to the other score that did perform similar to the phenotype. Did that answer your question?
Yeah, I guess I was just wondering if there was maybe genes that were included in one or not the other.
Yeah, there were differences definitely in the genes that were included, but examining that was beyond the scope of the work that I did. But that's definitely something to be examined in the future.
Great. Thank you very much for a great talk. Good questions.